Hello students, this is Dr. Yogesh Kumar, and today we are going to discuss about the process of recombinant DNA technology. So let us begin about our topic. As we know that the process of recombinant DNA technology involves several steps, and these steps has been divided into following 5 stages. The first step is isolation of DNA, fragmentation of DNA by restriction endonucleases. Second process will include isolation of desired DNA fragment, then ligation of the DNA fragment into a vector. Third step will include transferring the recombinant DNA into the host. And the fourth one will include culturing the host cells in a medium at large scale, and ultimately there will be the extraction of the desired product. Now, we move to our first step, that is the isolation of the genetic material, and that is about the DNA. As we know that in majority of the organisms, that genetic material is deoxyribonucleic acid or DNA. And in order to cut the DNA with a restriction enzyme, first of all, it should be in pure form, so that we can further process this DNA. And the first step to get the DNA is wherever it is bound. As we know that generally in a cell, this DNA that is found in the nucleus. Okay, so just to get that DNA from a cell, we have to lyse that cell. There are various type of cells, just like the bacteria, plant or animal tissue, or there may be any fungus also. Okay, so we use various enzymes to get destroyed or to remove all those membranes in which this DNA is protected. Okay, so in case of bacteria, we use lysozymes. In case of bacteria, we are using the lysozyme. And in case of plant cell, the outer layer that contains the cell wall contains the cellulose. So to remove that cellulose, we are using this enzyme cellulase. Similarly, the chitinase is used in case of fungus. So here what we are doing, first of all, we are removing the outer membrane. Okay, then the nucleus or the cellular material that will be in direct contact with the enzymes, and that DNA will come out. Next, let us see that all the genes, we know that they are located on the long molecule of DNA, and in that DNA also, actually here we can see that there are certain proteins in which it is bound. Just suppose I'm making here the histone proteins. Just this is the histone protein. Just I'm making only this is a histone protein. And this DNA that remains bound around this histone protein. So this is the DNA. So whatever DNA we have obtained after the fracturing the cell, the DNA will also not be in a pure form. So there will be impurity of protein. Okay, and certain RNA will also be present there. So here we see that there are at least 2 main impurities you will find in these DNA, and this extracted DNA, whatever that material is, actually that is being treated with other enzymes also, and these are the proteinases, and they are the RNases. The RNA will do, or RNAs will remove all the RNA which is present in that suspension. Similarly, there will be the protein. So the proteinases will digest the protein. Ultimately, we will get the pure DNA. Okay, and that pure DNA that is spooled with the help of chilled ethanol. Okay, here we can see that how it looks. Actually, here this is what this is the DNA, and this DNA looks like this after the treatment. Okay, now move to the next thing, and that is the cutting of DNA at specific locations. Actually, we know that all the DNA that is of that maybe or may not be of our use. Just suppose this is a big strand of DNA. Okay, and this big strand of DNA that contains 2 or 3 genes, that may have 2 or 3 genes. Just suppose this is a gene A, this is gene B, and this is gene C. Okay, so we should be aware that what specific gene we are required, and to cut that specific DNA segment or that specific gene, we are using enzymes. Those enzymes are known as restriction enzymes because they cut only at specific locations. Just suppose I have to make a cut, or we require this B segment or the B gene. Okay, so we will use the specific restriction enzyme to cut this B gene. Okay, so with the help of these specific restriction enzymes, we will be able to cut the DNA. Then there is a process, or we can say that to know that whether we have got our desired specific gene, we use gel electrophoresis to separate out that specific DNA segment. Actually, this gel electrophoresis is a machine, and that machine is having a positive charge and a negative charge. And as we know that the DNA is a negatively charged molecule, so it will move towards the anode or the positive charge of the gel electrophoresis machine. Okay, so that in that direction, this specific segment will reach. Okay, now the process of separation of DNA or that vector DNA is also done. Okay, and after when we have received both type of DNA fragments, or we can say that the DNA segment which is required, in which we have a specific gene, and that is to be attached to the vector DNA. So initially, these two, vector DNA as well as the desired DNA, they were treated with the same kind of restriction endonucleases. So now they are mixed with each other, and joining process is carried out. And the joining process of these two segments is made with the help of ligases. Okay, just suppose this was our DNA segment, which was B gene. This is to be introduced. Then this is our vector DNA. Okay, with the help of ligase, these two segments, they will get merged with each other. Okay. So just suppose this is the B gene. So this DNA fragment. That will be known as recombinant DNA. Okay. And as we know that if we have extracted a few number of desired gene, we can amplify the number of our interested gene. And for that purpose, we use a PCR reaction or polymerase chain reaction. Okay, in which what we do, actually multiple copies of our desired DNA is being produced. Okay, and how that is being produced, let us see. Just suppose this is the DNA. Okay, this is the DNA fragment that is to be amplified. In first step, what is being done? Actually, we know that two strands, they remain attached with each other by complementary sequencing. So I'm making complementary sequencing. Initially, what is done? That heating is done in this PCR so that both of the strands, they can separate from each other. And after the heating, these two segments, they will get separated. And after the separation, there is a process which is known as annealing. In annealing process, a small RNA segment, which is complementary, that is being attached. Okay, so just suppose here, this a small segment is being attached. So on both the strands, a small segment, preformed segment, that is being attached. And then the extension will occur. And that extension is occurred with the help of an enzyme that is known as DNA polymerase or Taq polymerase. With the help of this Taq polymerase or DNA polymerase, the extension will occur. Okay, the process of extension will occur, and complete two copies of the DNA will be formed. With the help of this PCR reaction, we can produce millions of copies of the desired DNA segment. Here we see that that if the process of replication of DNA is repeated many times, the segment of DNA can be amplified to approximately billions times. Okay, and such repeated amplification is achieved by the use of thermostable DNA polymerase. Actually, we know that when the heating process is used, and at the time, if the normal DNA polymerase is used, then that can denature, or that DNA polymerase will not work properly. So there is a specific bacterium, that is Thermus aquaticus, whose DNA polymerase is used, and that Thermus aquaticus bacteria, that remains at very high temperature, and the DNA polymerase of that bacterium, that is not destroyed at high temperature. For that purpose, we are using that Taq or Thermus aquaticus DNA polymerase, Taq polymerase. Okay. Now there is insertion of recombinant DNA into the host cell or organism. There are several methods of introducing the ligated DNA into the recipient cells. Actually, whatever the DNA, the recombinant DNA we have produced all outside, that is to be introduced in a specific kind of cells. And first of all, let us take about the E. coli. E. coli is a bacterium, and the desired DNA, what we have produced, that is to be introduced into the E. coli. What we are doing, the desired DNA segment that is introduced into the E. coli by modifying the environment in which those E. coli are present. There is a very simple strategy that is used, that first of all, the temperature of the medium is increased a bit high, so that the plasma membrane that becomes a bit liquid. So that DNA which we have produced, that gets inside into the cell, and then that medium temperature is cooled down, so that the DNA is taken up. And now the question arises, how we will come to know that whether the DNA or the desired DNA has been taken up. There is a very simple process, that some of the genes of the antibiotic resistance are also being introduced while we are attaching our desired DNA. Just suppose we take an example of the ampicillin. Certain genes which are producing ampicillin resistance, if they are being added with our desired DNA, and that vector is being introduced into the E. coli, then what will happen? The E. coli bacteria, they will become the ampicillin resistance also. So whenever the ampicillin is added into the medium, if the E. coli is not dying, that means they have become the recombinant one. So that is the process. And the ampicillin resistance in this case is called a selectable marker. Now, obtaining the foreign gene product. So our bacteria that has been transformed, now we will start getting the product, whatever gene we have introduced into the bacteria, we will get that product now. So by this process, we introduce our desired gene into the bacteria, or we can introduce desired gene in the plant as well as in animal cell also. Okay, so this is about RDT. Thank you very much.